Hi everyone, my name is Imran, and today I'm here to talk about my current ongoing thesis project, which is revolved around evolution of aging and epistasis. So, what is aging in evolutionary terms? It's basically a gradual deterioration of physiological function with increasing in age, which eventually leads to increasing mortality and decrease in reproductive success of individuals in a population. In like many organisms, we see a vast variety of different patterns of aging. And the thing is that why aging evolves, like why is it like so persistent in nature with these many patterns? If natural selection designs organisms for optimal survival and reproductive success, so why does evolution not prevent aging in the first place? And to answer that, there were like three prominent theories of aging. First is mutation accumulation, second is antagonistic pleiotropy, and third is disposable soma theory. But I'm only focused on mutation accumulation theory for this project. According to mutation accumulation theory, the force of natural selection actually declines with increasing age, especially after the age of maturity has been reached, because organisms reproduce and pass their genes to the next generation, to the offsprings, and selection becomes inefficient to get rid out of any deleterious mutations, kind of creating a selection shadow where selection is inefficient. So for a given genome, over generations, there will be a lot of accumulation of neutral mutations under mutation selection balance. And within a lifetime, some of these neutral mutations might become deleterious in the late age, and they will still persist in the population because of the presence of such selection shadow in the later ages. Now, there have been a couple of, actually lots of demographic and population genetics models describing how these kind of mutations can accumulate, and they have used the two indicators of aging. One is mortality and the second is fertility, also called as Hamilton's indicators of aging. And according to these indicators, if mortality rate keeps on increasing upward with the increasing age and fertility declining after a given age, you will always obtain a stable equilibrium of mutations which are early age neutral and late age deleterious. Then MA theory should make sense, right? Like you should see it quite often, like followed in many organisms, but that's not the case. There is very limited evidence in support of MA theory, because as you see in this first graph, there is deaccelerated mortality rate in many organisms, as that with the increasing age, after you hit the peak for mortality rate, it starts to decline, and which has not been considered in these classical models of aging. There are also other patterns of survivorship and mortality rate in many organisms, varying from plants to animals to any multiple different species, and they don't really follow the pattern MA theory predicts. So, and also one crucial thing is that what about non-additive interactions? Because these classical models only considered additivity among these late-age deleterious mutations, and there is also like some evidence of epistasis among age-specific mutations. And talking about epistasis, let's make it clear what epistasis is. So if you see that frequency of deleterious mutations increasing, there will be like a combined effect of all these deleterious mutations on the fitness. And if they are under additivity, then the fitness will decline linearly with the increasing frequency of deleterious mutations. But if there is synergistic epistasis present, then the observed effect of this deleterious mutation is much more higher than the expected effect under additivity. While antagonistic epistasis is where the observed effect is much lesser than what you would have expected under additivity, and the fitness kind of like slowing, like dampening, and being nonlinear in these cases of epistasis. Along with that, there have been latest evidences in MA theory, latest advances in MA theory, new theoretical insights, which is basically not new. It's been like 10 years, more than 10 years ago, where these people did consider epistatic interactions affecting age-specific mortality and fertility among these deleterious mutations. And what they found, the predictions, those predictions were dramatically different from the classical models that they were. If you consider epistatic interactions among late-age deleterious mutations, then you will never obtain a stable equilibria where this kind of late-age deleterious mutations can accumulate. So the question is there a role of epistasis in the evolution of aging? Because after this theoretical study, not much has been done in this field of MA theory. And to investigate this question, I'm using Drosophila melanogaster laboratory populations, which has been maintained for over 400 generations, selected for longer lifespan. And what we do is that we use mutation accumulation protocol on these laboratory flies, fruit flies, and allow accumulation of spontaneous deleterious mutations on a single haplotype. And the haplotype is chromosome X2 and 3 in these flies. So as you can see, these are like populations which are kept at discrete life cycle, and the ancestral population that we used was at like 4-week lifespan. And in this gray section, you can see is the gray section where the selection shadow starts. So it's being a discrete population, so we are able to see, we know for sure that selection shadow is after week 4. And over 400 generations, there have been, multiple neutral mutations that have probably accumulated in this population. What we do is that we isolate multiple haplotypes from these ancestral population and put it under a relaxed selection using clone generator females that we have, and bottleneck every single line to a one individual without any recombination being passed, recombination when a single individual is passing its haplotype to the offspring. And because of this relaxed selection, the selection shadow is present almost throughout the lifespan of the individual, and this will lead to accumulation of a lot of deleterious mutations throughout the multiple generations, like across multiple generations. And to control for these different degrees of deleterious mutations that will accumulate, we have 2 controls. The first is the control line where the life cycle is like 2 weeks, just like the MA lines. And here the selection shadow, as you can see, is starting at week 2. So because of that, any kind of late-age deleterious mutations can easily accumulate in the control lines, but the early-age mutations will be at much lower level. And we are keeping, reducing the amount of deleterious mutations, controlling for them by having an outbred population of just like population size of 70 without any recombination, which is basically hemiclonal, multiple copies of a given MA line, and keeping them separate from that. Another control is an aged control where the life cycle of these MA lines is 4 weeks, and this will overall allow us to control for any kind of early age as well as late age mutations to accumulate very, very slowly, while control lines have higher rate of late age deleterious mutations. And we have such 15 lines, and everything has like 1 MA line with 2 controls and 15 lines such. What we do is that with these lines, we are assessing male competitive fitness every 4 to 5 generation to measure age-specific fitness decline at early age as well as late age. Early age being week 2 and late age being week 4. For a given contest, we have 3 target males, 3 competitor males, and 3 marker females, and we measure the proportion of offsprings coming from the target males. What we expect is that in control lines, mostly late-age deleterious mutations will accumulate. So as you can see, this is the expected results after 30 generations, what we expect. The control lines will decline much more for the late age, while MS will overall decline for both early and late age. And age controls will be staying constant for both early and late age, since the mutation accumulation is at much lower level. And we are also going to investigate the age specificity of newly accumulated deleterious mutations. What if these newly accumulated deleterious mutations are inherently late age? Then in that case, we'll see that both aged controls as well as MS will have drastic decline in the late ages, late age fitnesses. And the last thing is the big question, whether is the decline in the age-specific fitness linear or non-linear. By doing these assays every 4 to 5 generations, and we assume that we have an idea that this amount of accumulation of deleterious mutation is happening at a constant rate in these populations throughout the generations, we'll be able to track down over generations whether this relative fitness decline is linear or non-linear for a given early age or late age, or probably both. And the current results is that we are only still at generation 20. We still have long way to go. Actually, not long way, just a short way. And what we have seen is that both in MA and controls, the late age, in all of the lines, the late age have declined overall, which kind of tells us, probably suggests that these spontaneous latest mutations that are accumulating are inherently late age, affecting the late age fitness. And if you track down the fitness decline over generations, then MAs are declining drastically for both early and late ages, while the late age is declining much more faster in all the lines compared to early ages. While controls and age controls, the early age fitness decline is not that significantly different. And the significance of this study is that classical models assumed additivity among deleterious mutations, so why stop there? We are using this meticulous MA approach to help us investigate the role of epistasis in the evolution of aging, especially seeing how epistasis is involved in both, not just like mutation accumulation theory, but also we can also see it in how it's involved in antagonistic pleiotropy, and whether epistasis is associated with any age-specific fitness or not. We have been able to find some early signs of age-specific fitness decline due to mutation accumulation. It's still an ongoing investigation, and we are not only doing the reproductive fitness assays, we are also doing desiccation assays where we try to see how early age desiccation and late age desiccation resistance is being affected by these mutations. It's because it's been suggested that desiccation resistance is mostly associated with longevity and mortality, so we are able to like infer some way through that. Anyway, overall, this entire project will give us novel insights into MA theory, a newer modified version, and predictions that will come out of it eventually. And I would like to thank Adam and Troy, my committee members, and all the lab members. So thanks so much. Yeah.
So this is a two-part question. The first one is just a clarification, and then the second one extended. For the clarification one, are you also measuring directly epistasis on mortality rates, or are you going only straight to fitness and measuring epistasis on
Epistasis, the thing with the mortality is that I really wanted to do it, but doing longevity assays, these fruit flies live for like 4 to 5, like about 4 months sometimes. And the thing is that doing it every 4 to 5 generation means every 2 months I'm doing these assays. So it's just like logistically a nightmare to do those. So I'm still planning to do those mortality checks at the end of 30 generation just to see if there is some effect of mutation accumulation on mortality.
The second one is less relevant, but I'll ask it anyway.
Yeah.
So for additivity and non-additivity on mortality specifically, one of the one of the discussed alternatives to additivity is proportional hazards, where it multiplies mortality rates instead of adding to them.
Yeah.
I've been going to ask if you looked at that, but since we can't measure mortality rate now anyway, that's fine. Is that something you plan to look at or are thinking about?
About multiplicavity and additivity, I'm gonna do these linear and non-linear fits on a, considering whether these interactions are gonna happen on a multiplicative level or additive level. So I'm gonna do these linear and non-linear fits on both the scales. So yeah. Any other questions? As of now, I have no clue what genes are accumulating, what kind of mutations are accumulating, but I'm still freezing these flies every 4 to 5 generations with reference as an inbreeding fly, and then crossing them with an inbred lines. And eventually, when I'm done with this project, someone else will like take up with those sequencing work and try to, correlate the fitness decline with the what kind of mutations that accumulated there. So we'll get even clearer picture of what's happening at sequence level. Okay, cool. Thank you.
So Thank you everyone for coming today. My name is Erica, and I'm really excited to talk to you about the evolution and regulation of decoupled dimorphic scaling patterns in ants. So scaling has been a pattern of interest for a long time. Some prominent examples include the transformation grids of Darcy Thompson's, where he made shape changes on a grid system to try and explain morphological evolution, and Julian Huxley's book on problems of relative growth, which explores scaling patterns. Now, the evolution of how certain traits scale to one another has generated an array of morphological novelties and has promoted adaptive radiations from the jaws of cichlid fish to the beaks of Darwin's finches and to the large-headed soldier casts in ants. The study of how traits scale to one another is known as allometry and can often be visualized as how some trait of interest changes in relation to body size. And so one pattern we see is when the trait changes proportionately with increases in body size in a one-to-one manner. So for example, the legs of the crab changing proportionally with the larger crab. Another pattern is when the trait of interest is disproportionately larger with increases in body size. So for instance, the pincer of the crab becoming disproportionately larger with increased body size. And then lastly, where the trait of interest is disproportionately smaller with increases in body size. So for instance, the proportion of the head becoming smaller as you go from baby to adult in humans. And so very quickly to introduce ants, they are social insects, and in some species there are worker and soldier ant castes. The smaller worker will typically perform more colony maintenance tasks like taking care of the young and foraging for food, whereas the soldier, on the other hand, plays more defensive and food processing roles for the colony using their disproportionately larger heads and mandibles, which makes them rather easy to spot. And so next I'd like to show you a scaling pattern example in a well-studied ant species, Camponotus floridanus. And so imaged here at the bottom, I have workers, and at the top, I have soldiers. Graphed here, I have scape length on the x-axis, which is a proxy for body size typically in these ants because it's been shown to change proportionately. And then on the y-axis, head width, which is a trait often used to classify which is a worker and a soldier. And so this is what is typically observed in ant species. Soldiers have traits that are larger than workers, and they follow the different scaling patterns that I explained in my intro. And so now when I'm collecting data for Phytolidentata, which is a species that has dimorphic workers and soldiers, an unexpected pattern revealed itself. And so on the x-axis again, I have scape length, which is the first antennal segment, which you can see more easily here. On the y, head width, soldiers are in red and workers are in blue. And so when we look at the y-axis, head width, the pattern of soldiers having larger heads and workers having smaller heads stays true. However, the scape completely messes up what we would normally expect. Instead of workers having smaller scapes and soldiers having larger scapes, we have a complete overlap in morphological variation. And so the scape can no longer be used as a proxy for body size here. Because for instance, if I were to pick a value of 2.9, I could not tell you whether it belonged to a worker or a soldier, suggesting that the scape length is decoupled from body size, meaning that there is no intercaste scaling present. However, when you look at the distribution, the scape length is increasing within each group and the ranges tightly match, suggesting that there's intracaste scaling present. Oopsie, sorry, intracaste scaling present. And so when you look, so for example, like the smallest worker and smallest soldier will have the same scape length, but also that the largest soldier and largest worker will have the same scape length. And so this was really puzzling to us, and so we were really curious to see if this was present in other ant species. And so to do so, we leverage the amazing resource called Phytoli in the New World, and in this textbook, there are morphological measurements on 626 Phytoli species, meaning that I can check what their scapes are like across the genus. And to do so for each species, I take their dimorphic scape ratio, which is the soldier scape divided by the worker scape length. And on a graph where we have ratio on the x-axis and number of species on the y, a ratio of 1 means that the soldiers and workers, again, have the same scape length. Greater than 1 means that the soldiers are larger than the workers, and then less than 1 would mean that the soldiers are smaller than the workers. And so I took the dimorphic scape ratio for 626 Phytoli species and created a frequency plot. And what we find is a dimorphic scape ratio that converges around 1. So in many species, the workers and the soldiers have the same scape length and is not only present in Phytolidentata, but in over 60 other ant species. Sorry, my animations. But we also, but interestingly enough, there are also the extremes of this distribution, where the soldier scapes are larger than the workers, which is what we would have expected for all ant species, but then also that there are soldiers with smaller escape lengths than workers, which is really intriguing. And so across the Phydolia genus, we have all the different combinations of escape length ratios between the workers and soldiers. And so if you will humor my Photoshop skills for a moment, I'd like to give a decoupled dimorphic scaling pattern analogy. And so if we were to return back to the previous example with the crabs, if this dimorphic if this decoupled dimorphic scaling pattern held true for their pincers, then you could have a big crab with a small claw, but you could also have a small crab with a big claw. And I know this looks ridiculous, but this is basically what this pattern is showing. And so the next thing that we were curious about is to see if this decoupled escape pattern was present in other independently evolved ant species that had soldier casts. And so we collected morphological data on 16 other species, and this is just a phylogeny to represent their evolutionary relationship to one another. And largely what we found was that for the majority of ants, they followed regular antennal scape kind of body, or scape to body allometry, except for one other species, Cephalotes varians, which also show the decoupling. And so this kind of suggests that this could potentially be the origin of decoupled scapes. And we have Shannon presenting a poster on Monday that goes more into detail about these different graphs and some of our ideas. And she's at poster board A16, so if you're interested, please check her out. Okay, and so the next major question we had was what mechanisms are regulating this decoupled dimorphic pattern? And so briefly, this is a schematic to explain worker and soldier development. And during their larval stage, there's a period of time where they can become either a worker or a soldier, meaning that they are bipotential. And so during this caste termination period, environmental factors such as nutrition and optimal social interactions will result in a developmental switch for either caste. And so this switch is mediated by a juvenile hormone, where an increase in JH results in the soldier trajectory and not enough results in the worker. And past this switch point, this is where caste-specific traits develop and differentiate. So for example, like the large heads in soldiers. However, the adult ants themselves also feed back into what caste an individual will become, where adult soldiers have a soldier inhibitory pheromone, which actually blocks soldier development. And so coming back to the antenna and the scapes, we were curious to see how manipulating some of these factors at certain developmental periods affected the allometry of the scape. And so first we did manipulations on the bipotential larvae during the caste determination period. And so we set up 4 different treatment groups where we manipulate the hormonal and social environments. And so we have larvae that are raised by only workers, larvae that are raised by only soldiers, and then larvae that are treated with acetone, and then larvae that are juvenile hormone or JH treated. So here I have head width on the Y and scape length on the x-axis, and in the gray is just like background wild type individuals, and the top one is soldiers and the bottom is workers. And so for the worker-raised acetone, we get regular workers with expected scape lengths. For worker-raised JH treated, we get soldiers, and so the JH is able to induce soldier identity with, again, expected scape lengths. But then for the worker-raised acetone, we get workers, but these workers are smaller than any of the wild type that we have, and also smaller than the red dots. And then finally, the soldier-raised JH treated. So we get workers again, and so the inhibitory pheromone that I talked about earlier was enough to block like the soldier identity, but it made workers that were like larger than the red and purple. And so the main takeaways are that the manipulations on the bipotential larvae were successful in manipulating caste identity, but in general, the scape remained decoupled. But notably, the soldier-raised acetone made individuals that were smaller than anything we've seen before. And so next, we decided to manipulate the social environment again. However, instead of on the bipotential larvae, we did it on soldier-destined larvae, which is during the caste differentiation period. And again, here we have head width on the Y, and for the first time, hind leg tibia on the X. And I haven't shown you any hind leg tibia graphs before, but at least in Phaidole, you'll just have to take my word that it's a proxy for body size in this species. And so we have background again in gray, and so the smaller dots, like to the bottom left, are the workers, and the top ones to the right are the soldiers. Okay, and so when we do soldier-destined worker-raised, we get regular soldiers, as seen in teal, that have expected hind leg tibias. But when we do the soldier-destined soldier-raised, which exposes these individuals to the inhibitory pheromone, it actually makes these novel individuals that do not normally exist in the colony. It instead actually makes these ants with the same body size as the workers, but with heads that are large that follow the soldier regression, which we'll call, I guess, microsoldiers for now. So when we look at the scapes for these groups, the soldier-destined worker-raised, again, makes regular soldiers with regular scape lengths. However, the soldier-destined soldier-raised makes microsoldiers with these scape lengths that follow the soldier regression. And so altogether, these series of experiments show that manipulations using juvenile hormone affect the caste identity, and the scapes remain decoupled. However, manipulations of the social environment at different developmental periods can actually generate novel head-to-body and scape-to-body allometry. Okay, and so now when it comes to the evolution and regulation of decoupled dimorphic scaling patterns, there's still so much more to explore and so many questions to ask. For example, when it comes to the antenna, why do soldiers and workers have evolved to have the same trait size? And also what factors are selecting and constraining its evolution? On a more macro scale across the genus Phaidole, how have these ratios evolved from species to species? And in a more general sense, like, are there other decoupled dimorphic traits out there? And then lastly, with a developmental point of view, how do traits become decoupled and coupled in the first place? And how do pheromones and hormones interact in such a way that they can change and affect trait-to-body allometry? And with that, I would like to thank everyone for coming, for the Rajakumar Lab, Helen and Shannon specifically for helping collect a lot of this data, the Moreau Lab for helping with museum collections, measurements and stuff like that, and then our funding agencies, specifically SSE for the Lewington Award, which helped fund a lot of this research. So thank you. Any questions? If not, that's okay too. Yes?
So when you're, I don't know that much about ants.
That's okay.
So workers typically raise the offspring, is that right?
Yes.
So when you're manipulating the social environment so that the soldiers raise the offspring, and then that changes like their, what do you think might be causing that difference? Like, do you think there's something in particular about that social environment that affects the allotment?
Absolutely. So maybe I just like breezed through it way too quickly, but these soldiers actually have this social inhibitory pheromone. So it's like a contact pheromone that's just like on their exoskeleton. And somehow these pheromones are able to affect the development in such a way that it blocks soldier development. So that's at an earlier stage, but when I tried to manipulate it at a later stage, it affected the allometry. And so it might not be like what actually occurs in nature, like regularly, like typically, like individuals don't get raised by like 100% soldiers, but it is a way to experimentally demonstrate in a really extreme setting, like how allometry is being kind of messed with and perturbed. If that makes sense.
Thank you.
Yes?
That was really interesting. How is it different when they're raising the queens? You looked into any of that kind of biology?
Yeah, so getting queen-destined individuals is actually really hard. It kind of only happens like once a year, and we can't really predict it, but that would be really interesting to look at for sure, to see like how it happens in the different casts and ants. Thank you.
Hi everyone. So is the enemy of my enemy my friend? This is essentially a question that my group has been thinking about a lot in recent years in the context of the host microbiome. So I'm going to be talking a little about some of the theory that we've been developing recently on this. So I think it's fair to say that we have a pretty good understanding of how hosts interact both ecologically and evolutionarily with their pathogens. I myself have spent quite a lot of my time thinking about host pathogen ecology and evolution, and even coevolution as well. Understanding how traits like resistance, infectivity, and virulence might evolve when we have these pairwise interactions between hosts and pathogens. Obviously, though, this is a gross simplification of reality. Hosts and pathogens don't just exist in isolation together. They interact with other hosts and pathogens and predators and prey and competitors and cooperators. And yet we have relatively little understanding about how these additional species interactions might mediate that focal interaction that you really care about. And I'm especially interested in microbes and thinking about how microbial interactions in the context of the host microbiome might mediate pathogen ecology and evolution. And I'm going to talk today a little bit about defensive symbionts. These I find to be especially interesting. I'm going to think about defensive symbionts as microbes that confer some degree of protection to their hosts against pathogens. And defensive symbionts are found throughout nature. There are lots and lots of different examples and different mechanisms by which they can confer protection to their hosts. So, for example, we can see things like immune-mediated competition. Things like Wolbachia can confer protection to, say, fruit flies against things like protozoans and arboviruses. We can also see things like interference competition, so maybe some productions of public goods like superoxides. These can confer protection as well. So, for example, strep pneumo can produce hydrogen peroxide, and this can help displace Staph aureus. We can also see things just, overlapping niches, so straight-up resource competition as well can help mediate protection to hosts. So lots and lots of different examples throughout nature of defensive symbiosis, and we should expect they've helped shape the ecology and evolution of their host and pathogen populations that they interact with. They also present a potential interesting avenue for biological control or biocontrol. So, for example, you might have a host infected by some pathogen or is potentially exposed to a pathogen. The idea here is that you want to introduce some friendly microbe, a defensive symbiont, that will hopefully either protect or clear the pathogen from that host or host population. And a lot of the work that's been done to date has mostly focused on that ecological short-term level, thinking about, okay, what happens if we introduce this biocontrol? Does it make things better? Less attention, I'd say, has been paid towards the evolutionary implications of those biocontrols. So, here indicated by these colors, the pathogen is supposed to be these circles, and the biocontrol is this other microbe. The pathogen might evolve in response to introducing a defensive symbiont. We should also expect that defensive symbionts might evolve as well over time. We could also expect that the host will evolve as well. So, the kinds of questions that we've been thinking about is, how do constituents of the host microbiome evolve, and how do microbiomes themselves affect pathogen evolution, and then ultimately what might be the implications for host populations. So we put together a range of different models that essentially take these putative, very, very simple host microbiomes where we have a host, a pathogen, and a defensive symbiont. And then we see ultimately what are the ecological and evolutionary outcomes when we have these three species together. So moving away from just that pairwise interaction between a host and a microbe to now think about these tripartite relationships. One of the cool things I think about these sort of tripartite systems is that they're context dependent. So, for example, if you take away one of these microbes, you can always get back to like a host pathogen scenario. So that defensive symbiont might only actually be a mutualist in the presence of the pathogen. So that might be because the defensive symbiont has some mild level of virulence itself. It might be that it's just a little bit costly to harbor. And this is where this idea of is the enemy of my enemy my friend comes from. Well, it all comes down to some sort of ecological context. There's also an evolutionary context dependence as well here, because all these microbes can evolve along this continuum between mutualism and parasitism as well. So things are neither fixed ecologically or evolutionarily. So if you're not familiar with these types of models, I'm not going to go into the underlying sort of mathematical details behind this, but essentially this is just a schematic of how we set up these models. So we have hosts split into these different classes depending on which kind of microbes they have. So we have hosts that have no microbes, hosts that have either this pathogenic or potentially protective microbe, and then we have hosts that have both of these microbes. And then we just think about the rates at which hosts change these states. So, for example, they might get colonized by a protective microbe, they could be infected by a pathogenic microbe, and they could then also have births and deaths of our hosts. And then if we're thinking about what role does defensive symbiosis play, well, depending on the mechanism of protection, it could affect, say, your rate or likelihood of getting infected by this pathogenic microbe, which I'll refer to as resistance, or it could essentially offset some of the costs of disease. So if you have both the pathogenic and protective microbe, then perhaps you have a lower death rate from this pathogenic microbe, and so that would be an example of tolerance. And so what we want to do, first of all, is just think about, okay, what happens if we introduce this defensive symbiont? How does it affect pathogen evolution depending on the underlying mechanism of protection? So, for example, here we've got our defensive symbiont and our host, they're non-evolving, their colors aren't changing, and this pathogen is evolving. That's what the colors changing means. And so this is some work led by my postdoc Cameron Smith and also an undergrad Scott Renegado, where they were looking at evolved virulence in a pathogen on the y-axis and then varying the strength of host protection conferred by this defensive symbiont, and then comparing this to the ancestral level of virulence. This ancestral level of virulence is kind of like the optimal level of virulence in the absence of the defensive symbiont, so we're kind of perturbing that system. And so one of the key sort of takeaways here is that regardless of the mechanism of protection, defensive symbionts always select for higher levels of virulence in a pathogen. Okay, so there is a slight difference in terms of the nature of, or kind of the direction of how strong the change in virulence is depending on the mechanism of protection. This is because with tolerance, we see this really sort of rapid increase in virulence as you increase the strength of tolerance. That's because tolerance is directly offsetting the costs of virulence for the pathogen. Whereas resistance, there's still an increase in virulence, but it's relatively lower, and that's because resistance is having this disease suppression effect. So the key takeaway there, though, is that there is always selection for higher virulence. That's really the key thing. We also see potentially that tolerance, when it is very high, can also drive pathogen diversification to relatively high and low virulence strains. So now this is, again, virulence on the y-axis, but time on the x-axis. So we see this divergence into this high and low virulence strain. Essentially, by introducing this tolerance-conferring symbiont, you create an additional niche. So those were just sort of what happens if the pathogen by itself evolves. Now if we think about coevolution between our pathogen and our symbiont, so now both the host is still non-evolving, but our two microbes are now co-evolving together. Now on our y-axis, we've still got pathogen virulence, and now we've got strength of host protection on the x-axis, but this is subject to evolution. And we introduce defensive symbionts at different initial levels of protection. That's what these different trajectories correspond to. And depending on any underlying trade-offs or costs of host protection that these microbes might incur, so for example, if they're having to produce superoxides, then we would expect this would come at some cost because this is a public good. Depending on the nature of those costs, we could see still this elevated pathogen virulence evolving, but also maybe some stable intermediate level of host protection. But if those costs are too high, then we'll end up seeing loss of host protection. But crucially, even though you've seen a loss of host protection, you still end up with this elevated virulence relative to your ancestral state. So this could be problematic because you've introduced maybe a biocontrol that loses its protective value, but then you still end up with elevated virulence compared to where you were before. So you've made things worse. We can also see this kind of potential here where, depending on the initial level of protection, you could end up seeing a loss of protection, or you could end up seeing protection increase indefinitely. And we can also see things like runaway selection. All of these essentially depend on the underlying details of the trade-off. So it kind of tells you that understanding something about the nature of any costs associated with protection is key in terms of determining the long-term outcomes. And then thinking about what does this ultimately mean for the host population, especially if you're thinking with this biocontrol hat on. If you have a defensive symbiont that confers tolerance to its host, we see that there is always a net cost in the long term. So even if you start off initially, say here, we've got relative population size for the host compared to the absence of the symbiont, you might initially start off with things being great. You introduce this symbiont, suppose this is an agricultural setting, you've increased your crop yield, it's great. In the long term, because of selection for higher virulence in the pathogen or loss of protection by the defensive symbiont, we end up seeing this trend towards a net cost. So there's never a scenario where we end up with things being better if our defensive symbiont confers tolerance. So this is a slight sort of word of warning. These are not evolutionarily robust biocontrols. So yeah, like I said, there's always this detrimental effect on the host population in the long term if your defensive symbionts confer tolerance. This is not necessarily true if you have conferred resistance. It can occur depending on the nature of those costs again, but it's also possible to go from a situation where you have a net cost to start with to a net benefit in the long term. So evolutionary robust biocontrols are more likely to take the form of symbionts that confer resistance than tolerance. Okay, so I'd say the key takeaway, therefore, is essentially be aware of unintended ecoevolution consequences of biocontrols. I'm certainly not against using biocontrols in principle, but I think we should be going in with our eyes open and thinking about, okay, if we're going to start manipulating systems by introducing friendly microbes, we need to understand a little bit more about the mechanisms involved and make sure we understand any trade-offs that might be involved as well, because we might be driving sort of worse outcomes in the long term, even if things are maybe beneficial in the short term. And then just a couple of sort of advertisements or announcements. Recruiting grad students at SFU. I moved there a couple of years ago. This is beautiful BC. If you are a master's student or if you know master's students who might be interested in doing a PhD looking at ecological or evolutionary modeling, please get in touch. And also on Monday, Aline McPherson, who is also at SFU, and we are running a symposium on the genetic basis of coevolution, thinking about everything from matching alleles to gene-for-gene models and beyond. With that, thanks for your attention. Just a quick sort of thank you to students and collaborators who've been involved in this project. These are the papers that sort of cover everything that I've talked about, and thanks to funders. And any questions? Thanks. Yes. Sorry, can you say a bit louder?
What would be the mechanism for selection for increased virulence in the presence of biocontrol agents? And the second question to that is, how is it different from something like, say, vaccination?
Okay, yeah, so I'll try and cover those as quickly as I can. So the question was, what is the sort of reason for essentially why we see elevated virulence, depending on the mechanism of protection, and why does this differ from vaccination? Hopefully I've summed those up correctly. So in the case of tolerance, it's because tolerance is offsetting any costs of virulence. So virulence is costly for pathogens, and so it's offsetting the cost. Resistance is a little bit more complicated, but it's more actually a competition effect here. So by introducing this other biocontrol agent, you're kind of taking hosts away from the pathogen, so it's essentially increasing competition, which increases the benefits of having a faster lifestyle and having higher virulence, basically. Why does this relate to vaccination? Yes, it does. In particular, the finding about tolerance is very similar to ideas about like leaky vaccines. And if you have a vaccine that confers say reduces disease severity but doesn't reduce onward transmission, you end up with similar patterns there. So it's not, some of these findings are analogous to things with other interventions. So great question, yeah. Okay, yes?
Did you run any models where there's no cost of virulence? Because I know it's kind of empirically there's pretty many studies.
Yeah, the problem, yeah, if you don't have any costs of virulence in these models, basically virulence just goes off to infinity, right? It like goes to any sort of maximum it has. So there has to be some cost of virulence, or rather, sorry, if you have, say, if there's no cost of virulence, sorry, it should go the other way. I'm thinking of transmission in my head. Yeah, if there's no cost of virulence, then virulence will just go to zero. So it's kind of, that's a sort of a trivial, like you basically don't need to do the models in that scenario because that's what will always happen, because you would always get selection against virulence if there's no cost associated with it. So typically, yeah, we have a cost there. I agree though that some of the evidence tends to be like a bit mixed in terms of where that comes from, yeah. Okay. I don't know if I was the last speaker or if someone else is coming up, but thanks everyone.